So, good morning and welcome to the afternoon session of the day. This afternoon is devoted to the presentation of the student of the Electronics and Telecommunication. The first speaker is Stefano Cattini, and please.
Good morning. Today I will present my research activity titled Laser Light Scattering Technique for Biomedical and Industrial Application. In such a field, I have developed two different research lines. One aimed to the development of a cell mixing interferometer exploiting a power feedback control, and the other devoted to ocular fundus analysis. In the first activity, I will present the principle of measurement, the proposed power feedback control, the performed calibration activities, and a case of study. Then I will move to the ocular fundus analysis. Once again, I will present the developed principle of measurement, the developed setup and the measurement analysis, the preliminary test, and the in vivo validation. The first activity have been devoted to demonstrate the feasibility of a self-mixing interferometer exploiting an automatic power feedback control. Actually, self-mixing interferometer are current controlled, thus requiring a thermostatic circuit. Our solution will allow to avoid this thermostatic circuit, thus providing advantages in terms of cost, size, and the dimension. The self-mixing interferometer could be thought as a conventional Microsoft interferometer. The Microsoft interferometer exploits the square law detection provided by photodetector. That is, the photogenerated current is proportional to the impinging optical intensity. As a result, if we split the monochromatic field provided by the source in two different paths, the reference beam and the probe beam, and then we add the two halves on the active area of the photodetector, the photogenerated current is proportional to the phase difference between the two fields, thus to the different path of the reference and probe beam. As a result, if one path is kept fixed, that is the reference beam, whereas the other can move, the actual photogenerated current is proportional to the displacement of the target. Despite the mathematical complexity involved in self-mixing effect, if the power re-entering the laser cavity is sufficiently low to satisfy the Barkhausen criteria for stability, the self-mixing interferometer could be wrongly thought as a conventional interferometer, where the probe field is the field emitted by the laser, whereas the reference beam is the field emitted to the rear face toward the monitor photodiode. Now I will present the proposed power feedback control. It consists of four main blocks. The first one is the automatic power feedback control that impose the lasing current in order to achieve the desired optical power. Since in our system the current provided by the monitor photodiode is exploited both to the automatic power control and for the analysis of the signal, photocurrent mirror has been exploited in order to provide such current to both the sides of the circuit. Then an analog processing network process the obtained signal, and the temperature monitor allow us to online monitoring the laser temperature. Experimental activities have been performed exploiting the reported setup. The core of the system is the automatic power control APC and analog processing network. This provides the lasing current to the laser and analyze the photogenerated current. Then the Doppler source have been obtained exploiting a refractive surface placed on a rotating disc of a motor chopper controlled system. Then the laser beam generated by the laser is splitted by the beam splitter beam, and the part of the beam is deflected toward the power meter or the optical signal analyzer, whereas the other beam is aimed onto the target, thus obtained the desired Doppler shift. The neutral density filter F allows us to achieve the low feedback level condition. The temperature monitor and the temperature controller allow us to impose the desired power to the system, and the digital signal analyzer analyze the signal output of the analog processing network, thus providing the Doppler band. Here a picture of the developer setup. It's possible to see the self-mixing laser diode system, the beam splitter B, the neutral density filter F, the motor chopper controlled system, the rotating disc, the optic head of the power meter, and the digital signal analyzer. Well, once developed the system, a few activities have been performed. First of all, we have calibrated our system. Then we have estimated the optimal working condition and the frequency response. Frequency response have been estimated with both electrical and optical tests. Now, the actual signal provided by the photocurrent monitor is due to the phase difference between the two impinging beam. Thus, the knowledge of the lasing frequency of the lasing wavelength is mandatory to avoid errors in the interpretation of the signal. Thus, we have analyzed wavelength variation induced by emitted optical power and temperature of the system. Thus, exploiting the system previously depicted, we have changed the emitted optical power and the lasing temperature, and we have acquired under the spectrum for each condition. The obtained results are reported in such a slide. It's possible to see how the lasing wavelength increases with increasing emitted optical power and laser temperature. And this is displayed in the left figure. Right figure display the uncertainty associated with the estimation of the wavelength. Now, the amplitude of the Doppler signal is due to the coherence degree of the impinging field. As a result, temperature and emitted optical power may determine different amplitude of the Doppler signal. Then we have analyzed such variation in order to exploit the better performances achievable at the actual laser temperature. The system is, the experimental setup is the same previously reported. We have changed the lasing current, thus the optical power emitted, and the temperature of the laser. For each point, we have acquired the electric spectrum of the VS signal, and this we have recorded such amplitude. The next slide shows the variation of the amplitude of the Doppler signal in relation relative to the actual laser power and temperature. As expected, at higher temperature and emitted optical power, the signal provide a very high, the system provide very high signal, obtaining results analogous or even better to the one reported in literature by other authors exploiting bulky thermostatic circuitry. As last activity, we have performed we have estimated the frequency response of our system. Two methods have been exploited. The first one rely on electrical simulation of the photodiode current. Thus, exploiting the decoupling capacitance, we have injected into the photocurrent mirror a current of known amplitude and frequency. Thus, monitoring the signal output due to the variation of the frequency of the injecting current, it has been possible to estimate the frequency response. Such activity neglects the intrinsic frequency response of the self-mixing effect. Thus, a second activity had been performed in order to validate the reported results. The second activity exploits the experimental setup previously reported. That is, we have, as we have seen, once set the emitted optical power and the laser temperature, the amplitude of the Doppler signal have to remain fixed despite to the actual frequency of the Doppler peak. Any deviation from such constant value could be related to the frequency response of our circuit. Thus, exploiting the automatic power control circuit and the temperature controlled, we have imposed different optical power and temperature. For each point, we have acquired several spectra, and then we have estimated the variation of the amplitude of the signal in relation to the frequency of the Doppler signal induced. The pictures report the comparison between the two different methods. The red line is represents the data obtained exploiting the electrical simulation. The blue square represents the optical test, the results of the optical test. The data have been normalized in order to minimize the mean square error between the normalized electric field response and the obtained optical frequency response. Since one point was out of the green data have been obtained neglecting the point out from the curve. C'è modo per vedere il tempo? Because maybe I have to skip a few slides if I am too late. No, because I have two arguments, and this is a part which could be eventually skipped if I... È più di metà. Okay, well, I skip some slide, and I will move to the conclusion. I know it was quite long. Then we have demonstrated the feasibility of an automatic power control system for self-mixing interferometry. This provide advantages in terms of cost, size, and weight. Moreover, the system have been... So I move to... because I have skipped some slides, and so some comments can't be done. I move to the next argument, that is the ocular fundus analysis. Then, once again, I will introduce the principle of measurement, the developed setup and measurement analysis, preliminary test, and the in vivo validation. This activity was aimed to identify a suitable endpoint for transpupillary thermotherapy treatment. TTT, transpupillary thermotherapy, is an effective treatment for several ocular diseases. However, up to now, a few studies have been carried out in order to determine a suitable endpoint, thus leading to several under or overexposure. Since overexposure results in local blindness and underexposure results in ineffective treatment, this constitutes a limitation to the TTT diffusion. Now, at the basis of our model, there's diffusing wave spectroscopy. The technique is analytically quite complex. Anyway, it could be understood considering the laser source as a source emitting n photon for unit time. Then, if we suppose to irradiate the sample and then collect the back diffused photons, if we have a static scatter, then we will reveal n photons for each time interval. On the contrary, if the irradiated photons encounter different scatter arrangement in each time, the detector will reveal different number of detected photons. Thus, from the analysis of the autocorrelation function of the revealed photon, it's possible to retrieve information about the dynamics of the scatterers. Since in ocular fundus the scatters dynamics is mainly due to blood flow and temperature, the analysis of the autocorrelation function leads us information about the scatterers dynamics and thus to temperature and blood flow variation. In ocular fundus, light propagation is quite complex. Neither the ballistic approach nor the diffusional approach may properly describe the photon propagation. Thus, we have proposed a two-step analysis. First of all, we wrongly suppose that photon propagation in ocular fundus in a ballistic way. That is, all the photons follow the same path. This simplification allows us to retrieve an estimation of the mean of the scatters dynamics from the analysis of the autocorrelation function. That is possible to estimate the mean square displacement from the analysis of the autocorrelation of the electric field autocorrelation function. Then, scatters dynamics in ocular fundus is mainly due to thermal motion and blood flow. Thus, thermal motion could be properly described in terms of Brownian motion, whereas capillary flow could be described in terms of a random flow. As a result, the mean square displacement of the ocular fundus scatters could be described as the superposition of Brownian motion and random flow. Well, now, even so, we have previously exploited a ballistic approach. The light propagation performs a quasi-diffusional propagation into the fundus layer. Thus, it's possible to relate the different delay region to different ocular region. That is, at long delay times, the autocorrelation function is mainly due to the photon that have a short path between the sample. And in our setup was the photon that have sampled the retinal region. On the contrary, at short delay time, all the revealed photon concur to the formation of the autocorrelation function. However, as we have seen before, most of the revealed photon have sampled the choroidal region. As a result, the short delay time could be roughly thought as due to the photon that have scattered choroidal scatters. Well, sort of putting all together, we have proposed a ballistic approach that allows us to retrieve information about the scatter's dynamics. Then we have considered the quasi-diffusional propagation of photons within the sample, and this have caused so the different delay region could be associated to different layer of the ocular fundus layer. Long delay region are associated with retinal scatters, whereas short delay regions have been associated with choroidal scatters. As a result, from the analysis of the mean square displacement within the two different delay regions, it's possible to estimate temperature and blood flow variation in the two different layers. Here I will report the developed optical setup. The light beam, the laser beam provided by the helium source is coupled to the ophthalmic microscope optics exploiting the lens L1 and the beam splitter BS. Thus, the beam is focused on the ocular fundus. Part of the injected photons are back diffused and then collected by the optics of the ophthalmic microscope, deflected by the beam splitter B, and then coupled to the single mode fiber by the lens L2. The so collected photon have been processed exploiting the single photon counting module and the digital correlator. Now, several experimental activities have been performed in order to validate the provided system. First of all, we perform several measurements on a mechanical eye model. Then we move to in vivo analysis. Today we report two activities, one related to the intraocular pressure increase, and the other related to the temperature increase. Well, in order, preliminary tests have been performed exploiting the reported mechanical eye model. It was composed of two separated regions. The first one at the top was aimed to simulate the retinal scatters dynamics, whereas the second region at the bottom was aimed to simulate the choroidal scatters behavior. Since the investigated retinal regions did not contain retinal vessel, the dynamics of the retinal scatters could be roughly approximated as a Brownian motion. Thus, this region was filled with a proper solution of intralipid. On the contrary, choroidal scatters display both thermal motion and flow. Thus, a flow has been induced exploiting a perfusion pump. This is the result of the first activity. Briefly, we have developed two different stimulus. In the first one, we have changed the flow in the choroidal region. As we can see, such changements have induced changement only in the short delay region. On the contrary, the second activity have been performed changing the intralipid concentration within the retinal sample. And as you can see, this affects only the long delay region. Thus, this preliminary test demonstrated that the system is able to retrieve the autocorrelation function from ocular fundus system. And moreover, that the proposed interpretation of the signal is correct. Now, I will report a few experimental in vivo activity performed on a pigmented rabbit. All measurements were acquired from the inferior parapapillary region and in a region far away from visible vessel. As a result, no retinal flow have been investigated. The first measure was aimed to increase the intraocular pressure. Such an increase is known to induce a ischemic effect on a choroidal vessel, thus reducing the blood flow in the choroids. On the contrary, the second activity was performed increasing the temperature, the ocular fundus temperature, by means of the therapeutic laser diode. Here I will present the results obtained from the first activity. The first figure is related to the autocorrelation function. We have acquired one autocorrelation function before the expertness of the overpressure by the Goldman contact lens, whereas the second is acquired during the intraocular pressure increase. As you can see, the intraocular pressure increase mostly change the short delay behavior. And the second figure is relative to the mean square displacement estimated from the autocorrelation function exploiting the ballistical approach previously reported. The data have been obtained analyzing the mean square displacement by means of the two delay region approach previously reported. The data reports choroidal blood flow reduction, thus totally agree with the supposed ischemic effect. The second activity have been performed by means of therapeutic laser diode, 200 milliwatt power and 4 millimeter diameter of treatment region. Then we have acquired a few measures before and after the treatment, the 16-second treatment. The first figure is relative to the dynamics of the retinal scatters. As you can see, the diffusion coefficient increase, thus showing the ability of the system to investigate retinal temperature increase. The second block is relative to the choroidal diffusion coefficient and mean square velocity. As you can see, the mean square velocity increase currently to a flow increase aimed to remove the excess of power due to the to the laser, whereas the reduction in the diffusion coefficient may be related to a stiffness of the retinal molecules. Concluding, measurements have demonstrated the potential of the system to acquire the autocorrelation function from the ocular fundus. We have demonstrated that the system is able to retrieve information about the reduction of the choroidal blood flow. And moreover, we have demonstrated that the system is able to investigate temperature variation. That's all. Thank you for the attention. Mi sono...
Thank you, Dr. Cattini. The presentation now is open for discussion, questions. Nothing. Excellent. I want just to say that this activity has bring to 3 publications on international paper, yes, and 1 is in press, and several presentations to conference and proceedings. Let's continue with the second speaker, that is who is